I'm a professor in State University of New York at Buffalo, Pharmaceutical Sciences. So actually, I'm working on the LC-MS-based bioanalysis, um, mainly on the protein area. So mainly my research actually encompasses two related but quite different areas. The first area is global proteomics. So we developed a number of label-free proteomics methods to enable large-scale bioanalysis on the global proteomics level. For example, quantification of thousands of proteins in a large scale, hundreds of samples. Another area is more focused on bioanalysis on the protein drug biotherapeutics, mostly antibody-based. In recent years, our lab developed a number of technologies to address the challenges in bioanalysis of antibody drugs. To address this, we developed a trapping micro LC-MS technology, which has high sensitivity like nano LC-MS, but high throughput and robustness like traditional high flow LC-MS. So the method can achieve low nanogram per mil sensitivity, but with really high throughput, several minutes per sample, and can run thousands of samples without stop. So we think it's a practical technology for the pharmaceutical industry analysis, large-scale analysis. And another technique we develop is for tissue analysis. For tissue analysis, you need to do two things right. Firstly, you need to get rid of the blood, and secondly, you need to extract your target efficiently and reproducibly from the tissue. So to address this, we developed a differential labeling method to get the perfect, just right perfusion of the sample. And also, we achieve efficient and reproducible extraction of the drug and the receptor or biomarker, whatever, from the tissue. And then another technology we developed is for the accurate analysis to improve the accuracy. So we call the hybrid calibration method. Yeah, actually, the challenges there are multiple dimensions to this. Right now, although LCMS looks like it's very promising alternative to ligand binding and other methods for the protein drug analysis, the reality is very challenging. The first of all is the sensitivity. So we always think LCMS is a very sensitive method for bioanalysis, but it's not the case for protein analysis for a number of reasons. Firstly, the protein drug, a big molecule, it poses a disadvantage for a molar-dependent method like LCMS. And secondly, it's very important to dilute the biological sample. So for small molecule analysis, we usually concentrate the sample. For the protein analysis, we do the other way. So that decreases the sensitivity. And most importantly, for protein analysis, the signal to noise ratio are usually pretty low because the chemical noise are pretty high. Yeah, for tissue analysis, there are multiple challenges. So right now, the first thing I would mention that for tissue analysis, it's really important and getting more and more important because of coming up of those new constructs in biotherapeutics. For example, bispecific antibodies, ADCs, and we need to figure out how much of drug actually in the tissue. And also in the tissue, we need to analyze the target because most of the constructs has tissue target. Not only the drug, but also the target need to be analyzed. So therefore, the first challenge is for tissue sample handling. So how to extract the drug out of tissue efficiently, reproducibly is the first challenge. And second challenge is sensitivity because the tissue levels are usually tens or hundreds times lower than circulating levels. So we need an ultra-sensitive method. And yet another issue is accuracy. So we really need to get a good accuracy for tissue analysis, but it's very difficult to evaluate. So I think in the near future, LCMS will be more popular in the field of antibody drug analysis for a number of reasons. Firstly, LCMS method, the method development is cheaper and quicker. So usually you can develop a method in a week as opposed to several months by ELISA method and other traditional method. And secondly, the LCMS can work in different matrices. It works in plasma tissues equally well, and also the LCMS can quantify not only the drug but also the receptors in the tissue. It's very difficult to use any other method to do this. So recently, when the biospecific antibody and the antibody drug conjugate come out, it's really important to do the tissue analysis, and LCMS is by far the best method for tissue analysis. And people need to figure out how much the target is still in the tissue during the course of treatment, and also need to figure out the biomarkers for drug effect and side effect. So, and the LCMS has a multiplexing ability to analyze everything in just one analysis. So I'm very optimistic with the LCMS technology in the near future in pharmaceutical area, both industry and academia.